Welcome to the NPTEL session of NPTEL course Introduction to Developmental Biology. The Let me first brief you on what the NPTEL PMI is. NPTEL PMI session is a problem-solving interaction-based session that is being offered by NPTEL in conjunction with the Prime Minister Research Fellows, in which a particular Prime Minister Research Fellow, such as me, is going to go through the particular week's problems along with the students and interact with them and help them clear the concepts about the videos of that week via solving the problems with the assignments that are of the current week. Obviously, we cannot go through the assignments of the current run of the course, current run of the course. So instead, we'll be going through the assignments from the previous year's course, and that is what we will be dealing with today. So essentially, these sessions will include us solving various MCQ questions based on the Introduction to Developmental Biology course, and then understanding the concept behind this question as well as solving them together interactively. The major reason for this session is the interaction-based learning that can be provided via these, as most of the NPTEL videos are online and do not have an interaction basis with the students that are taking the course. And that is why the NPTEL office has reached out and begun this program so that an interaction component can be introduced in this session. So, to start with, let me introduce myself. I am Anubhav Chatterjee, a Prime Minister Research Fellow under the supervision of Professor Jonaki Sen at the Developmental Biology Lab in the Department of Biological Sciences and Bioengineering, IIT Kanpur. As a PMRS Fellow, I have some deliverables to complete by the end of the year, so one of which includes teaching students outside of our institute. And this is how I am completing this session is part of my deliverables. So, in order to understand what exactly we are going to do in this session, let me introduce the point that we will be going through. First, I will present a particular question that was asked in the previous year's assignment of the current weeks, which is comparable to this particular of which is comparable to the current week's assignment, and we'll understand the concept behind the particular question using some slides, and then solve the particular question together. Today's session is a dry run of the entire course, and thus no students are allowed here, and it is just me recording the session on my laptop. But from the next session onward, that is from the next Sunday from 6 p.m. to 7 p.m., the students who have enrolled for the course and opted for these sessions can join the session and have a live interaction with me, and we can solve the various problems together. It is very important that you understand the interaction based learning in these sessions is what is important, and I will very much implore you to ask various questions and clear any doubts that you felt during the week via these sessions. So without more further ado, let us begin. So this is the week zero assignment from the last year, and this is more or less a prerequisite for the course, and we will go through it with a couple of slides each for each question. To start off, let us look at the first question. The first question is, which of the following is a true statement? Linnaeus proposed the five-pillar classification. Similarity among organisms in a taxonomic category decreases with increasing taxonomic hierarchy. Similarities among organisms in a taxonomic category increases with increasing taxonomic hierarchy. And Whitaker proposed the binomial nomenclature. So this is a particular information slide on this particular question. As we know, as we know, the hierarchy or the taxonomy of different organisms is divided as such, with the lowest rank being species, followed by genus, followed by family, order, class, phylum, kingdom, domain, and finally life. So the question asks us is that if we understand this classification on this, so let us move here, or let us move to the table on the right side. You can see that these are examples of various organisms, and we will as we and we will go through the particular ranks of classification one by one. So chimpanzee is included in the species. Has a name, troglodytes, and humans is obviously Homo sapiens, and you can see the species name is different from every one of them. As we move higher in the taxonomic hierarchy to the next slide, we can see that some of them have the same genus, that is fact, for both chimpanzee and bonobo. As we move further up the rank, we see the family of many of them is same, that is Hominidae. So the human, bonobo, and chimpanzee all fall under the Hominidae family. The order Primates further includes the lemurs into this category, followed by Mammalia, Chordate, and finally Animalia, which also includes earthworm in this classification, which includes humans, bonobos, and chimpanzees. So it is obvious as we are moving higher and higher among the ranks, the diversity among the organisms are increasing, and at the highest rank of kingdom, earthworms and humans are included in the same taxonomic category. And thus, if we move back to the question, it is obvious to say that the two statements among the four options is the similarities among organisms in a taxonomic category decreases with increasing taxonomic hierarchy. That is, similarity between the organisms is decreasing, and as we can see, as we rise higher and higher up the hierarchy, as humans and earthworms, as we just saw, they categorized under the same kingdom, Animalia, but their similarities is very very low. The similarity between them is very low. But this is what the question is asking, and obviously the second answer is the correct. Next, let us move on to the next question. It is a match the following kind of question in which we are asked to match the left side with the right side. On the left side, we have the option Kingdom Monera, blue-green algae, diatom, and protozoans. And on the right side, we have primitive relative of animals, photosynthetic single-celled eukaryotes, cyanobacteria, and bacteria. And there are various matching options, and we will try to match them. So we know that Whittaker's five-pillar classification, in which Whittaker has been able to divide organisms into five separate kingdoms, which includes the kingdom protists, kingdom monera, kingdom fungi, kingdom of plants, and kingdom of animals. So here we have a comparison table for all the kingdoms. The kingdoms Prokaryota or Monera include mostly in unicellular organisms which have cell walls made of peptidoglycan, do not have nuclear envelopes or any closed enveloped membranous structures, well-formed membranous structures either, are autotrophic and can also be heterotrophic and include examples such as bacteria and cyanobacteria or blue-green bacteria. The kingdom protists includes unicellular and multicellular organisms where we can see cell walls present in some of the organisms, and they can also be autotrophic and heterotrophic. And these also contain nuclear envelopes, and examples of these are amoeba, algae, and slime leaves. Fungi are obvious, have mycelium composed of hyphae, and yeast. The example we have cite the example of yeast as a unicellular fungi. Obviously, the bread mold that we see is also fungi, as well as mushrooms. They have cell walls made of chitin and have well-formed nuclear envelopes. They have large pulpurulent vacuoles, are heterotrophic in nature. And examples of these include molds and mold fungi. Plants are obviously fall under the kingdom plantae, are multicellular and have a branching body, are not compact, have well-defined nuclear envelope, may and have cell walls made of cellulose, are autotrophic in nature, that is, they can generate energy from sunlight or natural sources. And there are various examples of plants. Next is the kingdom Animalia, in which the organisms are multicellular and most have a compact body. There is a well-defined nucleus and nuclear envelope. They do not have a cell wall, have small vacuoles, are heterotrophic in nature, and the examples include jellyfish, corals, worms, humans, everything. So when we go back to the question, after looking at it, we see that the kingdom Monera or the kingdom Prokaryote, as was in the table, is essentially includes the various bacteria and primitive organisms that are present or prokaryotes that are present. So kingdom Monera obviously can be connected to bacteria. Blue-green algae is something that we just saw as an example under kingdom Proteus, kingdom Monera. Yeah, under kingdom Prokaryota. And blue-green algae is essentially cyanobacteria. So B can be connected to 3. Protozoans fall under the kingdom Protista and are primitive relatives of animals. And finally, we have diatoms, which are essentially photosynthetic single cell eukaryotes. So we have the match, according to the match the following, we have A as 4, B as 3, C as 2, and D as 1, which is the third option that is given right here. Next, we move on to in the next question, which is which cell organelle contains a lot of hydrolytic enzymes? The options are endoplasmic reticulum, lysosome, Golgi bodies, and central backbone. So let us go through these organelles one after another. First, we see the endoplasmic reticulum. The endoplasmic reticulum is a network of tubular and flat structures with, which has a surface area of 30 to 40 times larger than the cell membrane. The endoplasmic reticulum is divided into two parts, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. With the rough part, which has the rough texture, because ribosomes are attached on it. The function of endoplasmic reticulum is essentially to synthesize proteins and lipids, which are the proteins and the lipids, and depending on which part of the ER is producing the proteins and the lipids, it is divided into granular and agranular ER. So the granular or rough ER is responsible for protein synthesis, and the agranular or smooth ER is responsible for lipid synthesis. Next, we look at the Golgi apparatus. The Golgi apparatus is known as the packaging center of the cell. It has a membrane similar to smooth ER, and what it does is it creates transport vesicles to deliver cargo from proteins and various other substances uh, reach the Golgi apparatus from the ER using transport vesicles, and the Golgi apparatus processes these cargoes to form lysosomes and secretory vesicles. So the Golgi essentially forms various lysosomes and secretory vesicles in which proteins are packaged and then targeted to various parts of the cells as and when required. And thus Golgi is known as the packaging center of the cell. Let's move on to lysosomes. Lysosomes are the intracellular digestive systems of our body. The lysosomes digest or destroy damaged cellular structure food particles, and foreign unwanted materials. How they do plagiarism through this? Plagiarism through this via the hydrolase enzymes that are present inside of them, which can responsible for breaking down proteins, glycogen, and lipids. Thus, going back to our question, which cell organelle contains a lot of hydrolytic enzymes? Obviously, the answer is lysosome. Let's move on to the next question, which is which one of the following biomolecules is responsible for the bilinear nature of the cell membrane? So the options are triglycerides, phospholipids, cholesterol, and hydrophobic proteins. So in order to answer the question, let us look at the information slide for this question. It is the slide is based on cell membrane and the lipid bilayer. Here you can see a diagram of the lipid bilayer. A thin double-layered film of phospholipids is what is covers the entire cell, and this is known as the lipid bilayer. One end of the phospholipids is essentially hydrophilic, which is soluble in water, and the other is hydrophobic, which is soluble in fat. And they are arranged on the cell membrane in such a manner such that the hydrophilic parts of the phospholipids are already either faced towards the exterior of the cell or the interior of the cell, such that the hydrophobic parts of the two layers are facing each other. So here we can see this portion is the hydrophobic region of the cell membrane, in which the hydrophobic tails of the phospholipids are interacting, and the hydrophilic portion either place outside or inside the cell, both of which environments are suitable for the hydrophilic tail, as both of them contain water. The cell membrane also contains embedded glycoproteins, which are integral proteins, which can be integral proteins of peripheral proteins. There are also a number of membrane carbohydrates that are present throughout the cell, and this layer of carbohydrates are known as the cell glycocalyx. Thus, going back to our question, which one of the following biomolecules is responsible for the bilinear nature of the cell membrane? The answer obviously is phospholipids. Next, after dealing with a bit of cell biology, we will move on to molecular biology. So the question we are faced with is, according to the central dogma of molecular biology, which one of the following is not possible? Not possible. The first option is RNA-directed DNA synthesis. The second is DNA-directed RNA synthesis. The third is protein-directed RNA synthesis. And the fourth is RNA-directed protein synthesis. So let us look at it. So let me first present to you the central dogma, which obviously you would have some idea of. The central dogma is presented by Francis Crick it tells us that DNA is the carrier of genetic information in the cell, and it can be transcribed into RNA or mRNA, and thus, and in this process, the information travels from DNA to RNA via a process known as transcription. Following this, the mRNA is translated to the proteins or amino acid structure, and this process is known as translation. The central dogma also includes other types of transformations, which can other types of processes, which includes DNA replication, in which DNA is the template for its own synthesis, and in that way it maintains the genetic information inside the cell across generations. Reverse transcription, in which RNA can be transcribed to DNA, and there can also be RNA-based RNA synthesis as well. But the main important point of the central dogma is that once information has traveled to the protein form, it cannot travel back in any way uh, back up the chain, that is either to RNA or DNA. That is, once protein has been formed, the information has gone to the protein form and it cannot be transcribed or transmitted back to RNA or DNA. So, when we go back to our question, it is obvious that the third option is not possible, that is protein directed RNA synthesis. All other possibilities are present in the central dogma, which include RNA directed DNA synthesis, which is reverse transcription, DNA directed RNA synthesis, which is normal transcription, and RNA directed protein synthesis, which is translation. Next, we move on to the next question, which is in the process of replication of a linear chromosome, while one of the two DNA strands is copied continuously, the other strand is copied discontinuously, producing several small fragments that need to be ligated. Why is this? The options are DNA polymerase begins replication from only one end of the chromosome. The second option is DNA polymerase requires a RNA primer to begin replication. The third option is compact packing of DNA in nucleosomes makes continuous simultaneous copying of both strands impossible. And the final option is DNA polymerase catalyzes DNA synthesis only in the 5' to the 3' direction. In order to understand the particular question and solve it, we must first go and look into the replication bubble. This is a big slide and we'll go through it slowly. On this yellow part, we have the overview of the replication bubble as it offers. The replication bubble is the part of the DNA that is unwound, inside which the DNA replication is going on. DNA replication includes a lot of enzymes, which includes helicases, which unwind the DNA present at this the unwinding end of the DNA. DNA polymerase is the most important enzyme involved, which actually leads the polymerization of the formation of DNA-dependent DNA synthesis based on the DNA template from the old DNA. So here in the dark blue, we can see the old DNA, and in the light blue, we can see the synthesized strand. Here, the second enzyme is DNA polymerase. The third enzyme is primase that we will get back to, and there is another enzyme involved, which is the SSB protein, which is a single strand binding protein, which prevents the easy unbinding of easy rebinding of the DNA after the helices are separated into two strands. So one of the most important things that we have to understand here is that DNA polymerases always polymerize DNA from the 5' to the 3' direction. That is they can only add nucleotides and help synthesize a new strand in the 5' to the 3' direction. And that is where the entire problem lies. You see that when we focus on just half, just this half of the replication bubble, we'll be able to appreciate that there are in the oldest DNA there were two strands. One was the sense strand, that is the upper one, and the lower one is the antisense strand. The sense strand was arranged in a 5' to 3' direction. Thus, when the DNA polymerase is say moving from right to left, then for the cell strand, the DNA polymerase is essentially synthesizing DNA from the 5' to the 3' direction. As we can see right here, the nucleotides will get added to 3'. That is the direction in which DNA polymerase can synthesize DNA. Thus, that part is in this particular strand, which uses the sense strand as the template is known as the leading strand. And the leading strand is continuously synthesized from the 5' to the 3' direction. But in, and the synthesis always starts with the help of a primer. The DNA polymerase always needs a primer to which it can attach to in order to start the addition of nucleotides. This particular primer is the RNA primer generated by the enzyme primerase. But in the case of leading strand, there is only one primer required that is not shown. And it is shown here in the small yellow box with the red, small red portion. That is the RNA primer. That will be later removed and DNA nucleotide will be added instead. DNPT will be added instead. But for the lagging strand or the lower bottom strand that is being synthesized, we have to appreciate the problem that is occurring here. In the lower strand, in the lower strand of the antisense strand of the older DNA, the DNA was arranged from the 3' to the 5' end. And thus, when the DNA polymerase is moving along from the right to left direction, it is essentially moving in the opposite direction to which it can synthesize nucleotides from the lagging strand. That is, it is moving from the 5' direction, from the 3' direction to the 5' direction. And thus, it has to add DNA in the add DNA pieces in the opposite direction. And this is where is the problem occurs. What happens is that the primerase adds RNA primers to various portions of the antisense strand. And the DNA polymerase, as it starts adding smaller and smaller portion, as it starts adding DNA repeats to the primers, when it reaches the 5' end of another RNA primer, it stops adding the particular DNA. And thus we have a short chain of DNAs of short chain of oligonucleotides that get synthesized on the lagging strand. So these are shown in small blue colors, as you can see right here. Here is a small chain of nucleotides that has been formed, followed by this and followed by this one. And thus we have in the lagging strand discontinuous synthesis of oligonucleotides. Because every time the DNA polymerase tries to add nucleotide, it faces another RNA primer and stops synthesizing DNA, synthesizing the oligonucleotide right there. Later on, these RNA primers are removed and then DNTPs are added in their place, and all of the small oligonucleotides are ligated using another enzyme known as DNA ligase. The These thus in the lagging strand, there are small oligonucleotide stretches, and these are known as Okazaki fragments. So let us go through the whole overview of the application once more. First, what happens is that helices unwind the parental double helix, after which molecules of the single strand binding protein stabilize the unwound template strand, followed by the leading strand being continuously synthesized from the 5' to the 3' direction by the DNA polymerase 3. So there are various DNA polymerases depending on the productivity and various other functions. Next, the primase begins synthesis of RNA primers for various Okazaki fragments. When the DNA polymerase 3 is completing the synthesis of one fragment, it reaches the RNA primer of another fragment. And at that point, it will detach and begin adding DNA fragments, DNA nucleotides to the 3' end of the fifth fragment primer in the replication form. That is, while the DNA polymerase is moving along this direction, it will keep on adding. It will keep on adding nucleotides from the 5' to 3' direction while moving in this right to left direction. And once it is faced with an RNA molecule, it will skip the whole stretch of the RNA primers and move to the next RNA primer region and start adding from the 5 prime to the 3 prime position. And thus we continue as the DNA polymerase 1 then removes the RNA primers from the 5 prime end of the second fragment, replacing it with the DNA nucleotides that it adds one by one to the 3 prime end of the third fragment. The replacement of the last RNA nucleotide with the DNA leaves the sugar phosphate backbone with the 3,5 end. So right here in this portion, these two ends will not be ligated when the RNA is removed and DNA nucleotide is introduced. This portion needs to be ligated using the enzyme DNA ligase, and hence DNA ligase joins the 3 prime end of the second fragment to the 5 prime end of the first fragment. And thus the DNA replication continues. Thus, when we go back to the question, it is obvious that the second strand or the lagging strand is synthesized discontinuously because DNA synthesis by DNA polymerase that only occurs on the 5 prime to the 3 prime. Next, we move on to a question on genetics. Among the options given below, choose the best option to answer the following question. If the mother's blood group is B and the father's blood group is A, what will be the child's blood group? The options are B, AB, O, and any of the others. So let's look into it. The question is pretty straightforward. If the father's blood group is A, then the father's phenotype is obviously be either AA or AO. Similarly, if the mother's blood group is B, the mother's phenotype will be BB or BO. And we obviously know that in order to understand the genotype of the progeny, we have to draw a Punnett square. So we have the father's genotype here on the top and the mother's genotype on the bottom. And I have drawn here two Punnett squares, but essentially there should be four depending on the combination. So one with AA and BB, AA, BO, AO, BB and AO and BA. So this is one of the things that people sometimes miss while answering this question. In the ABO blood group system, if a person's blood group is A, then their genotype can either be AA or their genotype can also be AO. This is something that sometimes gets missed, and that is why we end up drawing lesser number of connects or we get wrong genotype of the progeny. So here I have only drawn two instead of the four because it more or less helps us answer the question, but essentially there should be four is what I'm trying to say. So for the first Punnett we see that the fourth progeny that will occur will have it can have the blood groups AB. So from if the father's genotype is AA and the mother's is BB, then all the blood children should have the blood group A. But instead, if the father's genotype is AO and the mother's genotype is BO, we can see right off the bat that the protein genotype can either be A, B, B, or A. So they can have a genotype A. They can have either genotype A, B, AB, and this is wrong right here. It should be OO. That is the fourth child should have genotype of O blood group. And thus, using the second Punnett square, it is obvious that given the question, the the particular child blood group can be any of them. That is B, AB, or O, and also can be E. That is the option should be B. Next, we move on to another question from based on nucleotide sequences. Which of the following will form a palinko when paired with the sequence AAGCTT. And we have four options from the nucleic acid sequences as options. So let us look into it. First, let us look into what are palindromic sequences. A palindromic sequence is a nucleic acid sequence in a double-stranded DNA or RNA molecule, whereby reading in a certain direction, that is 5' to 3' on one strand, the sequence is identical in the same direction on the complementary strand. That is, when we read from 5' to 3' on the same strand, it should be the same when we read in the 5' to the 3' direction on the antisense strand. Palindrome sequences often form halo structure in DNA or RNA. So, uh, for example, we have the particular sequence given in the question, AAGCTT, which is part of the same strand. Thus, in order to form a palindrome, the first thing that should be is that the sequence should be complementary to this sequence. So on the antisense strand, we have the sequence TTCGAA, and while reading from the 5' end, the two sequences should be same. And when we read the complementary strand sequence, we obviously get AAGCTT. That is, it is the same sequence just on a complementary strand, and all bases should be complementary. This is what it means by a palindromic sequence. Thus, the answer to the question is from the 5' to the 3' direction, AAGCTT. Obviously, we should not make the mistake and answering from PPCGAA, because nucleic acid sequences are always read from the 5' to the 3' direction. So do not. So the answer is obviously not PPCGAA, but instead AAGCTT, which is the number one option. So this was all I had for today. This was the dry run for the week 0 assignment from the Introduction to Developmental Biology course and a PMRF NPTEL problem solving session based on it. From the next week onward, these all these sessions will be open to NPTEL students who have enrolled for the course. Please make sure to turn up for these sessions. These will be weekly sessions from 6 to 7 on Sundays. Everyone is welcome. And even if you cannot join for whatever reason, you can always look back at the sessions when you go through my YouTube channel, where all the sessions will be recorded and uploaded. You can go through the particular recordings, look into the concepts, and whatever doubts you have, you can directly post on the NPTEL forum for the course that will be available to students that have already joined the course. So you can just post the query with the title PMRF session, and I will be there to answer your questions. You can also reach out via other ways as well, like such as commenting or emailing me as well. But the preferred way of communication is via the NPTEL forum. Again, I'm citing the point that from next week onwards, since the sessions are live, I will very much like everyone to join and interact with me while we solve all the questions. This will not only help in your understanding of the various concepts, but also help you in solving the assignments of that particular week as well. So with that, I thank you for listening to this particular video, and I hope to see every one of you next. Thank you.